Hi everyone, thanks so much for joining us today. My name's Jackie Carville, and to get started I'll be giving some background on the company and then going through several different molecular biology workflows found in the Lasergene package. As a little bit about us, DNASTAR is a global software company headquartered in Madison, Wisconsin. Our sales team, developers, and support network are all located here, so if you do have questions, we have the full support team on site to answer you, and then use your feedback to make our software even better for future releases. We were born out of the E. coli lab of Dr. Fred Blattner, professor of genetics at the University of Wisconsin, in 1984. We built a strong reputation with our early products during the Sanger sequencing years that continues today. This past year, we celebrated our 30th anniversary, and we are looking forward to many more successful years as this field continues to evolve. While still offering some of these classic sequence assembly and analysis tools, we have expanded into the realms of next-gen sequencing, supporting all major next-gen sequencing platforms. In addition, we also now offer software to assist in the work of structural biologists. DNASTAR products have long been established on the desktop computer, but they are additionally available on the Linux cluster or the new DNASTAR cloud. And as a quick note, our software is currently designed for research use only. So as we can see from this graph, DNASTAR is the leader in citations in peer-reviewed journals. For the past 30 years, more researchers have cited DNASTAR software in scientific journals than any other commercial sequence analysis software. And this really speaks to the level of trust that our customers have in our products. So in terms of our products, we do offer 3 suites for different areas of research. We are currently on Lasergene version 12.1 and plan on releasing version 12.2 shortly. We've had several significant enhancements to these suites recently since we moved to version 12. The Lasergene Genomic suite provides all of the software necessary for next-gen sequence assembly and analysis. With the release of Lasergene 12, several new workflows in Lasergene Genomic suite were introduced, including enhanced SNP prediction accuracy and new gene panel workflows. The Structural Biology suite provides tools for protein sequence and structure analysis. Our protein structure prediction program, NovaFold, complements the Structural Biology suite. And finally, we have our Evolution suite, which we'll be focused on for the rest of today's presentation. Recent enhancements to this suite include new sequence alignment methods and tools that were incorporated into the Unaligned Pro application, which I'll be showing you today. All of these suites can be purchased on Windows and Mac platforms, on the desktop computer, or on the DNASTAR cloud. So this Evolution suite is our suite for evolutionary biologists to work with sequence alignments. And the suite includes a variety of different applications, but today we'll be focusing on our Unaligned Pro, as I mentioned, Sigmund Pro, SeqBuilder, GeneQuest, and Primer Select applications. So to briefly go over our agenda for this presentation, we'll first cover multiple sequence alignments in Unaligned Pro, next look at Sanger sequence assembly and analysis, explore some gene discovery capabilities, and finally look at some virtual cloning and primer design functionality. So I'll get out of the PowerPoint here and open up the DNASTAR Navigator. The Navigator is a guide to all of the DNASTAR applications. In looking at the Navigator, you can view a summary of all the DNASTAR applications available to you. If we hover over any of these icons, we can see a nice window that comes up with a more in-depth description of each application, and then access to some additional resources such as videos, tutorials, online help, and even some demo data if you're working with a free trial. The Navigator is also integrated with our help system, which is really nice as it allows you to search the DNASTAR help across all of our applications. But with that being said, I'm going to go ahead and open up our first application for today, which is our Unaligned Pro application. This application was designed to help you streamline your multiple sequence alignments, easily aligning DNA, RNA, or amino acid sequences. We have 4 different alignment algorithms within Unaligned Pro, and these include the Clustal Omega, MAFT, Muscle, and MAUVE algorithms. So within this application today, I'm going to go ahead and show you a few different multiple sequence workflow capabilities. And I'm going to start by first showing you just how to conduct a basic alignment using MAFT, which is the most recent alignment algorithm added to Unaligned Pro. So we'll first want to go ahead and add some sequences into our project, which we can do by using the Add Sequences to Project tool. And here I'm going to go ahead and navigate to some alcohol dehydrogenase sequences from a variety of different species and open those up in the program. And after this is open, we can take a look at some of the views in the Unaligned Pro interface. So at the top of the screen is our overview, and we can see the sequences that we added displayed as these blocks with no gaps. And we can see that all of these blocks have different lengths, which are proportional to the ungapped length of the corresponding sequence. We can use the zoom sliders to zoom out of this, in and out of this tool, both horizontally and vertically. And we can also use this viewport to navigate across the sequence. And as I drag this viewport, we can see that all of the views in Unaligned Pro are integrated, as I am using that to scroll across our sequences view here at the bottom of the screen. But right now I'm going to go ahead and align these sequences using MAFT, and I can do so using some of these commands under the Align tool. So once aligned, we can see that this overview here has changed, and now we see all these as green blocks of the same length, and we can see that these blocks are now displaying gaps, as we can see by some of these gray sections within the blocks. So for any alignment that's done in Unaligned Pro, you can choose to analyze the alignment in several different ways. First, you can explore the distance matrix, which I can access from the tabs at the bottom of the screen. And this distance matrix details distances between the pairs of sequences. We can also choose to create phylogenetic trees, which we can do in a couple different ways. So the first is through this tab at the bottom of the screen, and we can create some trees right here within Unaligned Pro. And the second way is to do our one-click export to an external tree viewer, such as Figtree, for example. And I can select that and then use any of the styling or analysis options available within this application as well. Another important way to work with your sequences and your sequence alignments here in Unaligned Pro is through our Style panel over here on the right-hand section of the screen. So our Style panel helps you to adjust parameters in any of the different views here, doing things from basic font size, and colors adjustments, as well as even adjusting some of the parameters available for the metrics used to calculate your distance matrix. And one last analysis method I'd like to point out here is our Tracks panel. And our Tracks panel gives us different types of methods that we can apply to any of these sequences here. This would include some sequence logos, as I already have displayed on the bottom of the screen. We can also look at a consensus match histogram for this alignment. And we can even choose to expand out any of these sequences to look at features. We can add a gap fraction. Lots of nice different analysis methods we can apply to our sequences here. So since I aligned some amino acid sequences for this particular example, the different tracks that you can apply to these sequences varies depending on whether you're aligning amino acid sequences or nucleotide sequences. So I'll point out this tracks panel again in our next example so that you can see some of these additional tracks that you can choose to apply. Again, more information on any of these views that you're seeing here in Unaligned Pro can be found in our integrated DNASTAR help system or in our Unaligned Pro tutorials. But right now I'd like to move on and show you the results of a couple other types of projects that you can do in Unaligned Pro. So first, I'd like to show you how the app works with different multi-segment files. So I'll go ahead and align or open up this new project here. And multi-segment files are sequences that consist of more than one chromosome, contig, or fragment. So let's say that you'd like to string together the consensus sequences from all of the contigs in an alignment, or chromosomes in a genome, or exons in a gene. So if you simply added these sequences in as we did in the previous workflow, you're going to end up aligning these parts to one another. And instead, what we need to do is enter in the sequences using a different method to make sure the entire set of sequences in the multi-segment file will be treated as an end-to-end concatenation. So as you can see, I've already loaded in several of these multi-segment files for different species here. And I'm going to go ahead and load in one more just to show you how to do this properly. So for this particular workflow in working with these multi-segment files, we're going to want to use this Add Sequences End-to-End in Project tool. And if I select that, I'll go ahead and navigate to my data files that I'd like to use. So I have all these different exon groups here, and so I'm going to load in these last four exon groups for the rhesus. And we'll go ahead and name this group as well, and click OK. And then we can see that these multi-segment files have now been added in this this row here titled Rhesus. So this particular project just takes a minute or two to run, so I'm going to show you the completed project here in the next window. So I'm going to open down this overview so we can take a look. And here we can see in the overview again that now our gaps are shown in the multi-segment sequences. And again, we can go ahead and use any of the views available within Unaligned Pro to analyze this alignment as well. As I said before, since these are some nucleotide sequences, there are a couple new tracks that I've applied here to the sequences as well. So we can see the GC content and the translation track that you have the option to apply here. So one of the other alignment methods that is available in Unaligned Pro are MAUV alignments. These are specifically designed to align genomes. So I'm going to go ahead and open up a completed MAUV alignment project, and I'll show you what this looks like. So as you can see, this particular alignment looks very different from some of the other alignments that we worked with already today, as these genomic sequence alignments are displayed as locally collinear blocks. Now, these blocks represent multiply aligned sections of at least two of the current set of genomic sequences. So discontinuity in the numbering is displayed as a break between the different blocks. And some of these connecting lines that you see here also show the correspondence between blocks. If you notice any of the blocks hanging below the black line, as we see with this green block here, this indicates that the hanging blocks are on the opposite strand compared to the other blocks. So we can select any of these blocks and notice that the whole alignment reorients itself to focus on that particular block. And then the block that's selected is going to be shown here in the sequences view. This grayed out version of the sequences view, this is showing you the end of the sequences from the adjacent block. So I'm going to go ahead and show you we can, like any of the other alignments, scroll through our particular block to look at our sequences. And again, we do have the option to apply different styling methods and tracks as well for these. So finally, I'd just like to point out one other bit of functionality in Unaligned Pro, and that's our profile alignment functionality. And this allows you to add additional sequences to existing alignments or merge two alignments together. So if we open up a new project here, I'm going to go ahead and add some sequences back in, and I'll go back to working with some of those alcohol dehydrogenase sequences I was working with earlier. But this time, instead of adding in all 5, I'll just add in 3 of these. So now that I've had these added, I'm just going to quickly perform an alignment so I can align these using muscle. And then now I'm going to go ahead and add in my other 2 sequences. And once I've done that, we can see that there are some new views available to us here. So we have both the aligned overview as well as the unaligned overview, and the same with the sequences. We have the aligned sequences view and the unaligned sequences view. Once we've added in these new files to the project, we can see that a new button was activated up here in the toolbar, the Merge Unaligned Sequences into this document's existing alignment button. And I can go ahead and click that, and this opens up a screen with some parameters we can choose to adjust. I'll go ahead and click Align. And we can see now that all of these sequences have been merged into one project. Again, as I mentioned, we can also take two completed alignments, load one into the document, and then choose to merge another alignment into that existing alignment as well, using that same drop-down menu. So this wraps up my demonstration of Unaligned Pro. I'm going very quickly through all these workflows today, as the purpose of this presentation is just to give you a nice overview of some of our molecular biology workflows. But for any of these workflows that you do see today, we do have a full-length webinar available in our webinars library. So as I go through these different workflows, if anything is of interest to you, and you want to learn more about, say, sequence alignments in more detail, you can feel free to check out those hour-length webinars just on that particular workflow. But with that being said, I'm going to move along to our Sigmund Pro application. So Sigmund Pro is our application for contig assembly and visualization. And this can be used to visualize the results of next-gen sequence assemblies from our Sigmund Engine application, or for Sanger data assembly and analysis, as we'll explore today. So we can use Sigmund Pro to run a variety of different Sanger data alignment workflows, and these include reference-guided alignments, de novo assembly, and the ability to assemble in groups. So I'm going to go ahead and show you today how to perform a reference-guided Sanger alignment with some E. coli data, as well as some of the post-alignment analysis options. So to get started here with our reference-guided workflow, I'm first going to point out the two windows that are shown here on the Sigmund Pro screen. So we can see our unassembled sequences window here, as well as the contigs window over here to your right. So I'm going to do most of my work setting up my assembly over here in the unassembled sequences window. So to import data, you can just drag and drop your reads right into this unassembled sequences window. You can also use the Add Sequences button here. And I've already added in a few sequences here. I have about 151 sequences loaded into this project right now. This is just a small selection of reads. When I show you the final project in a minute, this has about 22,000 different ABI files in the total complete project. In addition to adding some of these reads here, I've also gone ahead and added in my reference sequence as well. And again, I can just drag and drop this right onto the screen. So I'm going to want to make sure that this is marked as our reference to differentiate it from all these other reads that we've loaded in. So I can select it and click the Mark Ref button. And we can see that this is now italicized, which indicates that that is our reference. So if you'd like to see the chromatogram for any of these reads, we can just double click on it to open that up, and then choose to scroll across and check out that chromatogram. We can also see quality scores for these simply by holding down and selecting the Q, and then we can see some of those quality scores. We can also choose to trim the ends of any of these reads. So I can select a section of reads I'm interested in trimming and then select the Trim Ends button. This opens up some options we have for those, so we can scan our selections. And now we can see some of the updated limits here for those particular scanned files. I can also choose if I'm interested to trim ends based on a vector. So I can choose to set a vector here for any of these reads if I've used that in my project. And finally, if I'd like to check out some of this pre-assembly trimming, I can also select Project, Trim Report, and see exactly what was done to any of these selected files here. I can also go ahead and open up any of these chromatograms after trimming, just to see where that was trimmed. And we can see this bar here indicating where that was trimmed based on quality. So now that I've finished setting up this particular project, I'm going to go ahead and show you the results. This is particular assembly with all 22,000 sequence files. It takes about a half an hour to complete. So to skip waiting for that, I'm just going to go ahead and open up the completed project. So the first thing I'd like to point out here in our completed project is that we have all of our contigs listed here within the contigs view, and we can see whether each read was added into a contig and to which contig this was added. So we can look at any of these contigs in our alignment view just by double-clicking on that particular contig of interest, and we can scroll across this alignment view to look at all these reads that went into the particular contig. We can also choose to explore the strategy view in Sigmund Pro for each contig, and I'll do that by selecting the strategy view here. I'm just going to zoom in on this so we can see it a little better. And in this particular strategy view, we can explore our coverage, again, look at some features, our reference sequence, and the direction of all these individual reads here. Both of these views are highly interactive with one another, so if I click on a certain section, it'll hop right to that point in the alignment view. So it's a nice way to navigate through these views as well. Again, if I'd like to see the chromatogram for any of these reads, I can use any of these twisty triangles to just open those up so we can check those out. And finally, in Sigmund Pro here, in terms of analysis, we can also choose to look at the SNP report. And to open that up, I'll select SNP report. And this is our report that allows you to quickly navigate to your areas of interest. As again, this is also interactive with both the alignment and strategy view. So we can see the summary of all the total SNPs in our project, as well as explore some filtering tools to narrow down on some of those SNPs of interest. So as I work through the SNP report, I can choose to either confirm or reject any of these SNPs of interest. And so if I choose to confirm these SNPs, reject these SNPs, and as you can see, if you look up in the alignment view as I'm doing that, when I click on any of these SNPs, it just hops right to that particular place. And as I'm doing this, our reference sequence is being auto-annotated so that you can use this reference with your SNPs of interest for future assembly projects. So this here concludes my segment on Sigmund Pro. So now I'm going to go ahead and move along to our GeneQuest application. So this is our GeneQuest application, and this particular app within Lasergene helps you to locate and annotate genes, regulatory elements, patterns, and structure in your primary DNA sequence. So to get started with gene finding, the first step is to go ahead and apply some methods to your sequence. So right now I have a C. elegans project open, and we can see the methods that we can apply listed over here in the left-hand panel of the screen here. So any of these methods can be applied to the project by simply dragging and dropping them onto the assay surface, so just like that. And we can see in the right-hand panel of the screen our legend, and this identifies each of the methods that were applied to your screen. These methods can also be reordered by either on the assay surface or using the legend simply by dragging and dropping as well. So really easy to use, pretty intuitive there. So for this particular project, I've already gone ahead and set up the different methods I'd like to work for. So I'm just going to quickly show you how to create some features from the methods that you choose to apply in your particular project. So in looking at this right now, I've applied some of these Borodovsky methods here to the project. And I can see that this particular peak in the Borodovsky graph lies entirely within this particular open reading frame. So this indicates to me that this area could likely be an exon here, and I want to mark this as such. So what I can do is select Edit, go to Location, and I'm just going to go ahead and type in my location that I'd like to annotate. Select OK. And that goes ahead and just highlights that particular region of interest. So next what I can do here is just click on the annotation tool over on the left. I can choose to quickly title that and indicate the segment name, and I'll select OK. And then we can see that we've now created a new feature and displayed it right on the assay surface. So I can go ahead and just through that quick and easy method, go ahead and continue to add some of these different gene segments as desired. And once I've finished adding any of my annotations of interest, I can corroborate those by conducting a BLAST search. And I can do this by selecting Net Search, BLAST Selection. And this opens up our BLAST query window where we can go ahead and select our BLAST program and the database we'd like to use for that. So this wraps up my brief overview of the GeneQuest application. So again, just some real easy methods to annotate your sequences and explore some different methods as applied to your sequences in that particular app. So with that being said, I'm going to go ahead and move on to our SeqBuilder application for some cloning and primer design functionality. So I'll go ahead and open that up here. And so SeqBuilder is our application for sequence editing and annotation, as well as some virtual cloning and primer design. So to start a new cloning project, I'm going to go ahead and select File, New Cloning Project. I'll go ahead and scroll up to find where that was added. So we've got our Untitled Cloning Project window here, and this contains our folders for our inserts, vectors, and eventually the clone that we create. So in this application today, I'm first going to give an overview of some of the virtual cloning methods available in SeqBuilder. So in addition to restriction enzyme cloning, which is the workflow I'll show you today, SeqBuilder also supports gateway cloning, directional topo cloning, and TA cloning. So I'll show you first how to do a quick restriction enzyme clone, and I'll go ahead and select the Inserts folder first, and then select File, Import Sequences to Project. Go ahead and select my sequence of interest. And this opens up right away within the cloning project window under the Inserts folder. So now I'm going to want to go ahead and add in my vector. And for this, I can either add a custom vector that I create or one from our catalog. So for this example, I'm going to show you how to use our cloning vectors catalog. And to access this, I can select File, Open. And I'm navigating here within some of the demo data that comes installed on your computer with any download of Lasergene. So I want to select my cloning vectors catalog here. And so once this opens, I can see that our cloning vectors catalog is organized here by the types of vectors that you can apply to your project. And so I'll want to select my vector from the restriction vectors folder here. I can go ahead and just copy that and then paste it right into my vectors folder. So you can see that now both my insert and my vector has been added to my project. So now that I've done that, I'm ready to go ahead and create my virtual clone. And so I'll do this by opening up first my insert sequence from the inserts vector. And from this particular window here, what I'm going to want to do is select the sequence I want to use as an insert by clicking on enzymes at either end of it. So here I'm going to select this ACC651 enzyme, and then shift-click on this APA1 enzyme as well. And what we can see up here at the top of the screen is that not only has the proper range of the sequence been selected, but also the enzymes as well. And it's important just to double check this because these enzymes need to be included, obviously, for these restriction enzyme clones to work. So now I'm going to go ahead to my toolbar and select Cloning, Copy Restriction Insert. And then now I'll go ahead and open up my vector from my project window as well. And again, from my vector, now I again want to select the restriction enzyme sites that I want to clone into. So I'll again click on this ACC651 and shift click on APA1, and that whole range has been selected. So now that I've done that, I'll go back up to my cloning tab and select Clone Restriction Insert. So this opens up our clone insert window here. And in the clone insert window, we can see the ends of the vector and the insert to see if they're compatible. And you'll know right away if they aren't compatible because these two checkboxes will not be checked and the clone button will not be active. So in that particular case, you can simulate modification of the overhangs by choosing to slide the arrows to adjust length, or using the fill-in or trim blunt buttons here as well. Since these are compatible, I will choose to name our clone and go ahead and select the clone button. So this right away quickly displays and creates our new clone. Our insert sequence is highlighted here, and the new clone will also now appear within our particular project window. So any of these images that you do create here in SeqBuilder can be exported into our GenVision program to create publication quality graphics. And again, for this particular example, we did use vectors from that cloning vector catalog, but there is functionality available within SeqBuilder to custom add your own vectors to a project, and then add these custom vectors into that cloning vector catalog for future use. So as I mentioned earlier with some of our other examples, we do have an entire webinar detailing all our different cloning methods that I'd encourage you to check out to explore some of this additional cloning functionality that we do have in this software suite. So with that, I'd like to wrap up the cloning segment of the presentation, and now I'll move on to discuss some of our primer design functionality. So this primer design functionality is some of the last workflows that I'll show you today. And because there are different types of primers to design, I'm going to show you how to do that in a few different ways. So we have primer design functionality in 3 different Lasergene applications. Our SeqBuilder here, our Primer Select, and Sigmund Pro application. And which application you choose ultimately depends on what kind of primer design project you're doing and what your primer design goals are. So again, SeqBuilder here is used for PCR primer design. Our Primer Select application is used for some advanced primer and probe design. And then Sigmund Pro is for sequencing primers to fill in gaps or low coverage in an assembly. So what I'm going to do here is just go ahead and I'm just going to close SeqBuilder out and start fresh with a new SeqBuilder application. I'll just kind of show you how to open that up quick from the Navigator. And we'll go ahead and open our sequence that we want to use here for this primer design project. So what I'm looking at here in SeqBuilder is the linear map view of this particular sequence. So if I go to this linear map view and I select this neomycin kanamycin resistance feature as my region of interest, I can now select priming, create primer pairs, and this opens up our primer pairs dialog. So I'm going to go ahead and expand the conditions in primer characteristics so you can see some of these parameters that are available to you for modification. And so as an example here, I'm going to go ahead and adjust my target temperature here to 55, and I'll go ahead and click OK. And we can see that SeqBuilder has chosen the best primer pair with the characteristics specified that lies exactly within the selected region and then displays it in the primer design view. And so by default, we're looking at the top strand primer first, but I can toggle between the bottom strand primer by using these buttons here at the top of the screen. So we can choose to edit our primers as desired as well. We can do this by adjusting the length of the primer, or even just simply by inducing mutations by pointing and typing in our options there as well. And we can kind of see how that affects the sequence there. So you can really easily work with some of these primer pairs until they've been modified to your particular satisfaction. So now that I've shown you how to do some quick PCR primers in SeqBuilder, I'm going to switch back to Sigmund Pro, which was the application we used earlier for our Sanger workflows, to show you how to create some sequencing primers to close gaps and fill in areas of low coverage. So I'll move back to our Sigmund Pro application. And we can see that here in Sigmund Pro, I've just loaded in 2 sequence files here. And I'm going to quickly just assemble that into a config. And then choose to open up that config in our strategy view here. So now I'm going to go ahead and select config, primer walk. And this opens up our primer walking dialog here. And so I'll want to choose to create these primers to improve my coverage, and I'll require clone coverage on this project. And select OK. And we can see that that generated our primer walking report here. So if I just double click on this first primer, we can see that that opens up the primers here in our alignment view. And with our usual integration, we can see these primers as the small lines here in our strategy view as well. And so finally, as the last workflow of today, I'm going to go ahead and introduce our primer select application, which is our application you can use for your advanced primer design projects, just because this has lots of different options available to you for your primer design. So I have a particular sequence opened here, and first I'm going to show you selecting conditions, primer characteristics, and this will show you some of the parameters that you can choose to adjust for your primers. And I'm going to go ahead and leave these at their default settings for now, but these are available for adjustment by you. So now to get started, I'll select conditions, primer locations. And here we can choose to restrict our locations by a few different options. And here I'm going to choose to restrict my locations by the upper and lower primer ranges. And so I'll just quickly type in my ranges of interest. And click OK. And now I'll see the paired green and red triangles in my document window, which mark the allowable ranges for upper and lower primers. So we can automatically now search for the best primer pairs by selecting Locate, PCR Primer Pairs. And we can now see a list of primer pair candidates with the best choices shown at the top of the window. So if we double click here on a primer pair, we can choose to look at a composition summary, which gives you some information on that particular primer pair, as well as our primer hairpins report. So again, we just have some really nice detailed information within the primer select application here. As in SeqBuilder, we can also choose to edit our primers. We can select Edit, work on Upper Primer, and this opens up this nice primer editing dialog here. Again, we can choose to adjust the length of the primer or quickly introduce primer mutations and work from there. So this wraps up my primer design workflows and actually all of the workflows I'd like to show you today. So before I finish, I'd like to point out a few of our support options available on our website. So go straight to the DNASTAR homepage here. And just wanted to point out that under the support tab, we do have many different resources for you. These include our video library here, where we have over 200 videos organized by topic. So if you're interested in any of the cloning workflows I mentioned today, we do have short videos on a variety of these cloning workflows. Same with primer design as well, and with many of the different workflows we looked at today. If you're working with our applications, you can also choose to sort these videos by each individual product. This is a good resource to help you learn our applications as well. Additionally, we also have some online tutorials and our online help as well. This also can help you get started, answer your questions, learn the workflows and the software. And finally, we also do have our DNASTAR blog where we do post different helpful articles. So I'd encourage you to subscribe to that as well. So that concludes my presentation today. If you have any questions, feel free to email me at webinars@dnastar.com. In addition, if you do have any requests for future webinars, you can also send those to me at the same email address. And finally, I'd encourage you to go back to our webinars page and check back for more future webinars soon, as we should have those scheduled.